All right, now let's consider genes that are linked. For this instance, I'm going to use our genes G and N again, but this time I'm going to put them on the same chromosome, which means that they are indeed linked. What's the consequence of this? Well, now during meiosis, the homologous chromosomes are still going to separate from one another, and the sister chromatids will still separate from one another, giving rise to 4 haploid gametes. But what will their genotypes be? If I look, I'll notice that capital G and capital N are in one cell, and lowercase g and lowercase n are in the other, both times 2 because the sister chromatids split as well as the homologous chromosomes. So I'm left with the genotypes capital G, capital N, lowercase g, lowercase n. So far so good, just like the example of unlinked genes. Here's the difference. With the unlinked genes, the principle of independent assortment allowed me to flip-flop my chromosomes so that I could change what capital G associated with. In the previous example, if we flip back real quick, we notice that in the second version, capital G went with lowercase n, and the lowercase g went with the capital N. This was different than what we had originally started with here. That was able to happen because of independent assortment, because the genes were found on separate chromosomes. In this example, no matter what I do to this pair of chromosomes, I can flip-flop their positions. Capital G is still staying with capital N, and lowercase g with lowercase n. So I see just these 2 gametes produced. Now, in reality, we need to take into account crossing over as well. So let's take a look at what that does. All right, so now with crossing over, I'm going to show crossing over as happening between just the N genes, so that lowercase n gets paired with capital G and lowercase g gets paired with capital N. This is with crossing over. Now these homologous pair are still going to segregate. The sister chromatids are still going to segregate, which means that in this cell I have a capital G still, but now I have a lowercase n. Over here I have lowercase g and a capital N. Each times 2 because those sister chromatids also segregate, so that my genotypes are capital G, lowercase n, lowercase g, capital N. Now, these original 2 gametes produced here are known as my parental gametes, because the segregation of chromosomes in these 2 cells directly reflects what the parent had. Capital G and capital N were on one chromosome, lowercase g and lowercase n were on the other chromosome. Because these gametes reflect that, they're considered the parentals. Now the other 2 gametes, here and here, do not look directly like the original parent. Capital G is paired with lowercase n, which is not found originally in the parent. Lowercase g is paired with capital N, not found on the original parent. Because these are the results of crossing over, these gametes are known as recombinants. If I were to cross these gametes with those of a parent who was homozygous recessive, so little g, little n, well, then I would truly be able to tell the different results of that cross, and they would be attributed to our original heterozygous parent here. That would allow us to then tell which of the offspring displayed the recombinant genotype, the recombinant phenotype, resulted from that parent crossing over, and which of the offspring look just like the parents because they are the result of the eggs or the sperm that did not display crossing. Now, if you look at this, one thing you will notice is that these 4 genetically distinct haploid gametes that were produced from crossing over also look very, very similar to the 4 that were originally produced on non-linked chromosomes. So how are you supposed to tell whether it's due to non-gene linkage on separate chromosomes or these 4 different genotypes? Are they due to crossing over unlinked chromosomes? It's a very good question, and in order to answer that, you have to look at the offspring and at the frequency that these different gametes, genotypes, and phenotypes appear in the offspring. So you would cross it, just like in a test cross, with your homozygous recessive in order to do that. For example, if I were to look at my offspring and I saw that there was, I'll write these numbers in green, there were 100 baby fruit flies. And out of that 100, about 26 of them looked like this, had capital G, capital N, which would mean that the offspring looks like the dominant traits, capital G and capital N. This one, let's say, had about 24 out of the 100 offspring. This one had about 22, and this one 27, or rather 28. So if we add all of these up, we get 100 offspring. They're pretty equal proportions, pretty much 1 to 1 to 1 to 1. Very even. That would indicate to us, because these were found in these fairly even ratios, that these two genes were unlinked, found on separate chromosomes. However, if we go to our second example, if I were to look at these and I saw out of my 100 babies that approximately 42 of them displayed the dominant genotypes for both, the dominant phenotype for both, and about, let's say, another, let's say 45 looked like this. So far, that's a total of 87 offspring. And then another 6 looked like this, and 7 like this. So out of my 100 offspring, most of them, the 45 and the 42, had the parental phenotypes, the parental genotypes. A very small number have these other two genotypes. Because they're unequal proportions, that suggests that these two smaller numbers here are due to a crossing over event. Otherwise, if these two genes were unlinked, I would expect to find them in an equal ratio of 1 to 1 to 1 to 1. Because I do not see that, little red flag should be going off in your head saying, aha, these are linked genes and crossing over has occurred to result in these two recombinant phenotypes, the two recombinant genotypes. And again, you can tell that just by looking at the numbers of offspring produced that display each of these different phenotypes. Now, for our intents and purposes, we're always going to be crossing a heterozygous with a homozygous recessive in order to do these kinds of calculations and determinations in this course.